We don't need more data. We need courage to interpret what we already have. Imagine this. You run 2 RNA-seq experiments. Same conditions, clean QC, strong statistics. And yet, the top genes overlap only partially. Pathways point in different directions. Predicted transcription factors change depending on the motif library. Nothing is wrong. But nothing is actually fully coherent. You know these moments, and instinct is predictable. Generate more data. More samples, more sequencing depth, more omics layers, more integration. It feels rational, but what if the bottleneck is not in the data? What if it's in interpretation? In our recent genome enhancer analysis of longevity-associated SNPs, we began with hundreds of mutated genes. We reconstructed enhancer modules, we identified key transcription factors like YY1, HNF4A or FOXO3. Then we traced the network upstream and we identified master regulators like PRKG1 and others. Not from correlations, not from enrichment alone, but from structure, from regulatory architecture, from upstream causality. Modern biology measures everything: transcriptomics, epigenomics, chromatin accessibility, single-cell states, and yet mechanistic clarity remains very rare. Gene regulation is not linear, it's correlative, combinatorial, context-dependent, feedback-driven. Expression change doesn't automatically reveal the driver. Statistical significance doesn't equal mechanism. Adding more data doesn't actually automatically create insight. Sometimes it actually multiplies ambiguity. For decades, we believed if we measure enough, understanding will follow. Today, we measure everything, but the central question remains: who drives the system? This is not only computational, it's actually epistemological. What counts as evidence for regulatory causality? Every AI model inherits the structure of its ground truths. If regulatory knowledge is shallow, AI inference will be shallow. If binding models are noisy, causality becomes probabilistic storytelling, sophisticated storytelling, but still storytelling. There is a fundamental difference. Descriptive biology measures change. Mechanistic biology explains change. Explanation requires curated, regulated infrastructure, validated TF-DNA interactions, experimentally confirmed binding sites, structured signaling to transcription relationships. Without that, upstream analysis becomes unstable, not because algorithms fail, because the knowledge layer is actually insufficient. So when your dataset conflict, when the prediction regulatory sites disagree, when additional omics layers generate more hypotheses instead of fewer, ask the harder question. Do we truly need more data? Or do we need deeper regulatory knowledge to interpret what we already have? In complex biological systems, the difference between noise and insights is often the regulatory layer you cannot yet see. If this resonates with you, click the first link below this video and briefly describe what you are working on. RNA-seq, ChIP-seq, methylation, GWAS studies, just few sentences. We'll examine it carefully and explore how curated regulated knowledge from transfac can help structure your analysis and maybe stabilize upstream inference and clarify the causality mechanism behind your data. And then we will arrange a Zoom meeting to examine it together in depth. Because in nonlinear biological systems, more data is rarely the decisive factor. Serious science desires serious interpretation. Let's move from accumulation to causality.